them or overexpress them and see their functions. So one thing we wanted to do in our lab is to see whether glucose is regulating any circular RNA and they have any role in the beta cell physiology. So to do that, what we can do? How I will find the circular RNA regulated by glucose? I need some help. जितना जल्दी बोलोगे खत्म करेंगे, लंच के लिए जाएंगे. नहीं बोलोगे टाइम लगेगा, है ना? कैसे आइडेंटिफाई करोगे सर्कुलर आरएनए? Sequencing, right? So NGS basically. So you take the RNA, you do the NGS and then find how many RNA circles are getting expressed in low glucose and in high glucose condition. Beta cells are treated with low glucose or high glucose, and we are seeing how many circular RNAs are changing between this. Like you see here, there are some which are specifically expressed in low glucose, some are specifically expressed in high glucose, some are common which are like differentially expressed, right? So as I said, RNA sequencing, you know all how RNA sequencing work. I think we have discussed few times now. So you basically have the genome, then your RNA, you have fragments, because in the NGS, like I said, like you can read 400 million, but those are not very long reads. It is like 150 or 100 base reads. You cannot go 500 base reading, right? So you fragment the RNA, add the adapters, put it into the flow cell, then the flow cell reads like 400 million reads at a time, and you get all these reads and then align with your genome and then see which fragments are aligning, and based on this you can say, okay, fragment A is variant A is expressed in condition A and variant B is expressed in condition B, and this is all based on the alignment of the reads. But when you go for the circular RNA, we don't look at the direct alignment, because direct alignment with the genome are the linear reads. So those are coming from the linear mRNA, not from the circular RNA. So which read you will consider? The reads which are not matching with the linear transcriptome or the splice junction. So you have linear reads which are matching perfectly with the linear transcriptome or the genome, and you have fusion reads which are coming from exon-exon boundaries or from RNA circles or any kind of misarranged chromosomal relocation. Okay, so you have one fragment from one chromosome, another fragment from another chromosome. That way you will have fusion reads. When you align with the genome, you will see, oh, half of the fragment is coming from this region, another half is coming from other chromosome. And in case of circular RNA, what you see is the first half of the read. Here you see the RNA-seq read. This is the first half. First half of the read matching with the downstream sequence, and the last half of the read matching with the upstream sequence. So it's a head to tail joining. So basically all the reads are like split into different halves and matched with the genome, and if the last half is matching at the upstream sequence, first half matching with the downstream sequence, then you can see this is might be coming from the circular RNA or of the head tail joining. And then the program can tell you, okay, these 3 exons are there, so joining those 3 exon sequences you can get the full sequence of the circular RNA, right? All got it? Yes or no? हाँ, 4 लोग ही यस बोल रहे हो. हाँ, बाकी लोगों को भूख लग रहा है? हाँ, नहीं समझ में आया? हाँ, स्किप कर दें? नहीं. Okay, so let's see. So this is your GIAN course at Guru Ghasidas Vishwavidyalaya, Bilaspur. Imagine that is your transcript sequence or your genome sequence. You want to do RNA-seq. Now you fragment that, put the adapters, go for sequencing, and then align with your reference genome. Now you have multiple fragments here aligning at different places. You can say, okay, this is my mRNA and perfectly matching. But there is a fragment called Vishwavidyalaya Guru. Okay, that is not there in your reference. Right, either Guru is matching or Vishwavidyalaya is matching, half is not matching. Okay, in this case, like you said, first half is matching towards the end and the last. This could happen only if Guru, right? You can read this from